My name is Bhodi Tims. I'm the program director for the Masters in Herbal Product Design and the Post-Baccalaureate Certificate in Cannabis Science. And we're going to talk about ginseng today, which is one of those endlessly fascinating herbs, right? Complex in many ways, rich in culture as well as science. And the things that really I want you to just take away and maybe think about as we go through this is the number of factors that are required to delineate the clinical outcome for something plant like ginseng. What role the medical system plays in providing context, medical systems being everything from native to TCM to what we call Western, which is a polyglot. And that means really all the types of extractions, the plant species used, the dosing. What ecological influences are there on the therapeutic endpoints? What are we aware of? And can we find some convergence between all of these factors so that we get some clarity in terms of how and when and in what way we can use ginseng? So there are lots of different species out there, and I'm sure I have missed some here, but we're going to focus on Panax ginseng and Panax cankifolia. American or Chinese ginseng is how I'll refer to it. And so we'll start where, because of most of us are rooted in the Western context of U.S. traditions, and that means traditional native medicine. And a lot of this came from Dan Mormon's work. The general use is a stimulant, headaches, ingestion, infertilities. You can see that there are a number of different ways that it got used within tribal cultures. And some of these, I think, are like the Meskwaki women using it to obtain a husband. I'm not sure that was actually ingested. Maybe it was traded because it had such economic value. But it's been used as a love charm in other places. And I think some of what we're seeing here is a misunderstanding of the language use and the cultural idioms on how people were using it. So this is what's come to us in sort of Western ways. I don't have the data because I think it's verbally communicated on how a lot of tribes actually use it. But we do see this plant showing up early in the U.S. There are a number of different traditions. There are physio-medicalists. They were using it as a nervine tonic and relaxant. And they, even at that point, were aware of brain function effects. The eclectics, who really built a very strong written tradition. A lot of contemporary herbalists were trained in eclectics or folks that learned from eclectics. And they were used to calm nervous dyspepsia, indigestion, stomach problems, and nervous exhaustion. And particularly long-term tonic for patients with depleted resources. This is something we'll see in the TCM-based approach as well, that people seem to learn early on that when you had constitutional depletion, that this is really an excellent herb to turn to. And the root actually was listed in the U.S. Pharmacopoeia starting in 1842 as a medicinal plant. So now I'm not going to go deep into different traditions because frankly, there are probably a number of folks in the herbal community that could do more around this. I have some background in this, but not enough to call myself a TCM practitioner. John has got some experience with TCM. If he wants to jump in, he's welcome to. So we're looking at Chinese ginseng, which is Panax ginseng. And now these phrases come from TCM, and they have very specific meanings that if you haven't ever studied it, it may sound like a sensory or organoleptic signature. But in very different ways, TCM ties together meridians, qualities, actions in a very unique manner. This is a very old system, and it's quite effective. So here we're looking at trying to highlight maybe some of the differences that we'll see in the Chinese and the American ginseng in TCM, and those are the bolded elements. And we'll see also one of the ways that this was designated energetically was that the plant itself came from the north, and the north having qualities of cold, and therefore um, a denser quality about it. And that's part of how the energetics got formed. You see this plant being used with older populations and with constitutional debility. The American ginseng in TCM, Panax quinquefolia, rather than being warm, is considered cold. And rather than focusing on spleen, we're looking at kidney. These are not organ functions, but rather meridian lines and functional energetics in TCM. Here we see a lot of tonification going on. The other thing, this came from the southern Cantonese ports and therefore was associated with the south, and therefore it had energetics associated with the south, warmer. And here we see the difference in that this is nourishing yin, which is a regulatory process. It's calming and balancing. And the use, instead of with older folks, is really more contemporarily around urban populations, fast-paced lifestyles, because it's less stimulating than Chinese ginseng and therefore more appropriate for daily use, and also in both sexes. And so what we're not clear about is how much the role of male females shows up in TCM in terms of how they're valued, because in many respects, ginseng wasn't really offered to women. Now, whether that was an energetic difference or sexism, I'm not clear. I don't have that answer. I see JW typing a bunch of symbols. I don't know if you were trying to um, speak out and say something, but let me know. In the West, we look at ginseng really as an adaptogen. I gotcha. Your cell phone on your keyboard. I got it. Western adaptogens. Here we're looking at the hypothalamic pituitary adrenal axis. So we're looking at harmonization or amplifying hormonal regulation in a very coordinated system. And we'll see in a minute how the hormonal system is linked to the CNS, the central nervous system, in many respects. Panosian, who's done a lot of research out of Russia on adaptogens, noted that these adaptogens as a whole group, not just ginseng, worked on a cellular level to prevent cortisol-induced mitochondrial dysfunction. So again, we're looking at something that influences energetic function. But we'll see later that there's a lot of CSN-based qualities to herbs from Western science perspective. So one of my teachers was David Winston, and this is someone that has actually incorporated and integrated TCM with Western and native traditions. And he has a very unique understanding of a lot of these herbs that fall in all the different pots. And for him, American ginseng was really indicated for around stress, nervous exhaustion. We're looking at immune impairment, chronic colds showing up, bronchitis, tiredness, chronic fatigue syndrome. And so for him too, this was a cooler ginseng. And the Chinese ginseng was considered much warmer. That's what we call white ginseng in most instances. Red ginseng is more often out of Korea. It's steamed and considered very deeply nourishing and very, very hot. So this is some of the energetic differential that we'll see. And we're looking at immune functions, cortisol production, overactive stress responses. It tends to have a stronger impact on actual performance, mental and physical. And we're seeing this being used in older or deficient people suffering from exhaustion, depression, and or adrenal insufficiency. So again, this seems to be more deeply embedded in sort of constitutional aging processes. And so there are some real confounding factors when we try to begin to talk about that. In the US, ginseng products usually are correctly labeled genus, but there's a lot of variability in the concentration of what we call marker compounds. Marker compounds can often be the actives, but in many instances, we select 4 compounds that we think may be the active, and we latch onto them for quality assurance purposes to measure their presence as they go through processing, extraction, bottling, storage. And so it's a quality assurance factor and may or may not be an active. but we see great variability in those compounds. And so the suggestion is that standardization should be really necessary. Let me know, I've got a mower coming past outside, if that's interfering at all with the sound. So I think this last line probably is true of almost all herbals. More complete characterization of herbal products should be considered. Thanks. Appreciate that, Gene. So some of the other confounding factors. You've got phytochemical differences. There are about 81 ginsenosides that are shared among Chinese ginseng, red ginseng, and American ginseng. And when those compounds get normalized, sort of measured as a ratio with a precursor ginsenoside, R of O, 3 groups emerge. So they do a statistical analysis, and they see that there are 3 distinct groups depending on the distribution of those ginsenosides. There are many that are in common, but there are many that are unique to the white ginseng, red, and American ginseng. And that the ginsenoside profiles are more similar between the red and the white, which is sort of Korean and Chinese. And you would expect that because both are Panax ginseng, but significantly different from the American ginseng. So for those of you that aren't deeply embedded in phytochemistry, I'm just going to talk about this briefly. And I'm going to ask you just to hold for a second here. I've got my little puppy chasing a bowl that had tuna fish in it across the floor. It sounded like a rock and roll concert. So these ginsenosides, this mentions 30 plus ginsenosides. That's probably fairly standard for each of these. They don't contain all 81. And they're referred to in the literature as triterpene glycosides, steroid glycosides, saponins. And so these are all true. It is a triterpene. It's a steroid molecule. If you look down at the picture at the bottom right, you'll see pointing to the A-glycone, the non-sugar component, which are those 4 rings linked together. That's called a steroid backbone. It tends to hold that molecule fairly rigid, and oftentimes that rigidity is associated with specific cell receptor binding. And saponins refer to the soapy quality. If you ever had ginseng extract and you've shaken it, it gets very bubbly and soapy. It's one of the QA processes you can use to sort of check your ginseng extracts. And each of these ginsenosides, they differ by the type of sugars, or what we call the glycone, and the number of sugars that are attached, as well as where they attach. And you can see that little image down below, the glycone. Those are 2 sugars hanging off. And we'll see later on that they can actually hang off from different parts of this molecule. And this does 2 things. It affects how the ginsenoside gets absorbed, how quickly or how slowly it gets absorbed, which has a clinical endpoint, and also the effect that it has. So these ginseng, the ginsenosides, actually, we would consider those prodrugs because orally ingested ginsenosides actually go through the stomach and the small intestines without decomposition. But they get into the colon, and the bacteria there actually cleaves off that sugar from the parent structure, what we would call protopanaxidiols or protopanaxithiols. Sorry, that should be thiol, not triol. And I'll show you what those look like. And we can look at these as both a quality assurance molecule, but also to some extent it can hint at its use clinically. So when we look at these diols and thiols, what we're really seeing is this R2 and R3. Those are side chains. And you can see down below, we have ginsenosides that are labeled RB1, RB2, RB3, ginsenosides on the other side, RE, R1, and the R2 and R1s, or the R3s and the R1s, are placements on the molecule, and what you see below is a designation of a type of sugar. And these are all slightly different sugars, or if it says H, it's no sugar at all. And again, the presence of those sugars influences their how quickly they get into our blood, and to some extent their clinical endpoint. This also is influenced by the gut cleaving them off and leaving behind a molecule now that's activated. So when we look at some of the differential now, we can use some of that knowledge about the ginsenoside structures and say, well, where do they show up? Right? Where are we getting them in different types of ginseng? And in this brief graph or table, you can look at it in two ways. If you compare the P. quinquefolia in the blue with the P. ginseng, you can see that in the American ginseng, the quinquefolia, the saponins that are dominant are RB1 and RD, and in ginseng, it's RG1. In one instance, it's a diol, the other instance, it's a thiol. And so that gives us a little bit of a hint on how we might use these plants differently. And then when we look at wild versus cultivated, because this has been a big issue that I'll give you some more data around in a minute, is wild source versus cultivated ginsengs, and that they're different medicinally. And in this case, if we look at the distribution in the wild versus cultivated, we see a similar pattern in that the wild tends to have higher levels of RB1s, RB2s, and the cultivated RG1s. And so this says a cultivated ginseng is going to have a different clinical impact than a wild ginseng. Sorry about that. She sees me on this computer and she wants my attention. That's a Riley Rose being a wild thing. My apologies. So the greatest chemotype variation, and when we say chemotype, meaning the chemical signature, phytochemical signature, we see that in wild populations, which makes sense because what we have going on is all kinds of conversation between the plant and its surrounding ecosystem, fungi, bacteria, other plants. This is referred to as chemical ecology, which is really the major focus of my own system, my own research over time. And the thing that we're seeing is that these woods grown versus cultivated ginseng have different neuronal activities in terms of inhibition or stimulation, and that the differences can be traced back to actually wild or what occurs in the woods versus what's cultivated. And that can be traced back to the dominant saponins or protopotaxidiols versus thiols. So here are some central nervous system effects, right? RG1 and RB1 both enhance CNS activities, but in different ways. This RG1, and remember the distribution, we saw more of RG1 in Chinese and the woods grown, tends to increase nerve growth, protect nerve cells from damage. And that higher content in the Chinese ginseng stimulates central nervous system, impacts wound healing and hypoglycemic effect. It's considered hotter. Now, this does tend to mimic the TCM-based approach, right? Because the RB1, which we saw in American ginseng and wild grown, tends to exert a tranquilizing effect. This tends to slow the uptake of neurotransmitters like GABA, glutamate, dopamine, norepinephrine, serotonin, tends to stabilize the GABA receptors. And the higher content in that quinquefolium or American tends to calm the CNS system and is considered cooler. So this is where different cultures have come to the similar characterization about endpoint use and the nature of those herbal extracts, right? In both instances, designating one hot, one cool. And some of the interesting linkage that we see is between the central nervous system and the hormonal system, the endocrine system. So the endocrine system glands are rooted in the nervous system, hypothalamus and pituitary. The adrenal medulla actually derives from the same base cells that produce nervous system ganglia. And both systems communicate by the same neurotransmitters. So you get norepinephrine, which functions as a neurotranslator and an adrenal hormone in the endocrine system. And so both these systems also maintain homeostasis. It looks to balance out the response. And in doing so, it tends to respond to communication that's internal from within the body, as well as external from outside the body. So one of the things we see when we talk about that adaptogenic response, right? We saw that both from Panosian and David Winston earlier. These adrenal glands tend to secrete catecholamines. These are the epinephrines, the dopamines in response to stress. They have the same effect on the target organs as a direct stimulation by the sympathetic nerves, right? So all these responses, right? Increased rate and force of heart contraction, constriction of blood vessels, dilation of bronchioles, all those things. What does that look like to you, right? To me, that looks like the flight or fight response. And we see that in that direct sympathetic nervous stimulation. So these adrenal and nervous system responses can look very similar. So in research from Hasegawa, the in vitro model was they used chromaffin cells from the adrenal gland. This is also present in the sympathetic nervous system. These are some of the precursor cells we were discussing. These cells produce catecholamines, right? We discussed what they were, the norepinephrines. These are the systematic transmitters, right? Acetylcholine is a neurotransmitter. So ginsenoside metabolites actually inhibit the secretion of catecholamines from cells that are stimulated by acetylcholine. Normally they would get in and they would trigger that flight or fight type response. Now, if you think about one of the ways that adaptogens get sort of characterized in contemporary language is that they limit the metabolic cost of the biological system responding to stressors, external stressors, internal stressors. So this could be exercise. This could be you're going to school and you are under high stress. This could be environmental stresses. But in instance, it tends to limit the biological cost of those stressors. It maintains homeostasis without going to extremes. And this is in essence what we're describing here on a cellular level. But the thing about it, this metabolism of ginsenosides in the gut is required for this effect. So that reinforces that idea that the ginsenosides are prodrugs, that they need to be altered through digestion for them to actually be more active. Question from Maria. You said, can we assume that American ginseng would be better as a substitute for benzodiazepines? Yeah, I mean, really, why are you using the benzodiazepines? That's what I would go to first. What's the indicator for you to look at that? Is this a response to stress? Is this an adrenal function issue? Is this depression? So that's where I would start. And then if you can trace that back to the types of calming effects that you see in American ginseng, that's the reason for having them, because there are probably some uses for benzodiazepine which are masking as opposed to actually altering the systematic imbalance that's occurring, right? So I think benzodiazepines have actually helped some people. Movement disorder disease and benzos act to control it. Okay, so you're talking about some spastic muscular contraction and relaxation, I'm assuming. Yeah, I think probably the American ginseng. So I'm not going to get into too much clinical diagnosis, but American ginseng in combination with some other herbs would be useful. And we'll get to the idea of formulation at the end. Okay. So when we talk about product design, the questions I ask are, what are we going to learn from traditional knowledge, right? And do we need to consider the effect of growing conditions? These are two elements, right? And I would include in that also processing and species used, et cetera, things that we've discussed already. But here we can see, and this is something we do on our programs, something called gap analysis, where you look at where tradition points you, and when you can, you try to find coalescent information or data from science, because that helps strengthen the potential that that idea, that strategy actually will work. When you can't find that, then you have to find other ways of linking the two bodies of knowledge, because it turns out there's an awful lot of linkage. So one of the things we looked at was dosing, right? TCM dosages tend to be much larger. They tend to be 3 to 9 grams of powdered root. The Western tends to be a standardized extract, like a tincture, and usually about 1 gram. Now the saponins, these is another name for the ginsenosides. Their concentration is about 70 milligrams per gram in ginseng, right? And the standardized extract, we don't really know what the ginsenoside ratios are. So what we do is we get clinical trials in Asia reporting findings that are markedly different from the West. And that's because the East is actually using larger doses, but less standardized. So they actually have different ratios of compounds just based on standardization. And standardization is the process of trying to concentrate some of those marker compounds, getting more of those in, thinking that those are the actives, and we want more of that. And that can be useful in particular situations. Other situations it can work against. The other thing is we're getting different ratios just based on TCM, often using different species. So when we try to look at dosing, you really have to burrow down and be pretty explicit about what plant they're talking about, what form was used, and what the actual amount was given. And then if you're looking at Western, how did they standardize? What were they looking for as a filter? So some of the impacts of dosing is we've got stimulating effects on the central nervous system. When we, and this is set aside the differences in the Chinese versus American. In both instances, if you use very low doses of both herbs, you tend to get stimulating impacts more from the Chinese. And then very high doses, you tend to get inhibiting responses. Again, the inhibition or the calming element is greater in the American than the Chinese. And this is something we refer to as biphasic dosing or hormesis. And so as you go into studying these herbs and how they might be used, you want to be highly aware of what the data, what the traditions say about doses. So whole plant extracts too actually improve the oral absorption of these ginsenosides. So when they look at an equivalent dose of, say, the RB1 and RG1, and they use those as isolates, pure ginsenosides, and they have, say, 100 mgs of each, and you get a whole plant extract that has 100 mgs of each in it, as well as all the other ginsenosides and compounds that are extracted, you actually get higher oral absorption with the whole plant extract, because there are things going on that influence what we refer to as the pharmacokinetics, what the body does to these external exogenous chemicals that are being ingested. And the chemical ecology too. I find this interesting because we'll see in a minute some of the actual TCM-based extraction actually remove some of the highest concentration plant parts in terms of ginsenosides. The highest concentrations are in root hairs. And so in this picture, those little white antennae that stick out at 10, 2, and 6, these are called root hairs. And as you move in from there to the interior taproot, right, which is that green area, the pericycle, you're getting less amounts of ginsenosides. So you have more in those root hairs. And as you age, and I'll show you some data on that, as plants age, They tend to increase the ginsenosides up to a certain point. And they also decline after mid-September. Now, that last piece tells you that these ginsenosides in the root actually play a role in the environmental communication, right? Because everything goes quiet, quiescent in the regions of China and the US where this plant grows after September. So the need for that compound to be around to actually influence some of the exterior communication and relationships doesn't exist. We see this in studies in New Zealand as well. Now, this I found interesting because you're taking the plant out of its normal ecosystem, putting it somewhere else altogether. So I wanted to bring this as a comparative. The fine roots, again, those root hairs contain the most abundant ginsenosides. And for both, the concentration of the RB1 was highest in the underground parts. In ginsenoside, RE was highest in the above ground part. So we can actually use this RB1-RE ratio as a QA marker, meaning that you can get compounds that are powdered that get mixed leaf with root and called it all root powder. And to some extent, you can discern it by the color, but they can also use bleaching agents which change colors. But you should have a standardized RB1 to RE ratio for pure roots. That would be very different than if you threw in a bunch of leaf material. So here's some data on the aging, and that red box shows you that for almost every ginsenoside, it tends to max out at 16 years, excuse me, rather than aging even further. And so this says something about the optimal level. Now, what I've never seen and what would be fascinating to me would be to look at some of the differential clinical effects. If you were looking at a 5-year product versus a 16-year product, and you use them over multiple clinical conditions so that you could get a varied response and find out, is one really better suited for a particular clinical endpoint than the other? Not that one is better than the other, but they might have very different uses based on this variability in the ratios. Now, you could go through and say, are the ratios different? There's certainly, the levels are greater in the 16-year, but I think if you look at it, you'd find that the ratios are actually different because you're seeing sometimes a 2.5 times versus a 1.5 times greater amounts in the 16-year. So you're actually seeing an actual fingerprint that looks different. So some of the economics that influence quality assurance issues, we look at the current market value. And so the current market value in wild American ginseng is $700 to $900 a pound. And this is almost all purchased by the Chinese and exported. Young wild American ginseng, which is 4 years or less, $250 to $350 a pound. Woods cultivated American, $200 to $300, and you can see the rest. So you can see how this is going to influence any number of things. In one instance, you might find greater adulteration in the wild American because it's so valuable. And we can see that this is a trend over time. You get what I would refer to as commodity-like pricing spikes, right? This is natural for most agricultural commodities, that you get a variability in the pricing depending on supply and demand. But what's happening, and you see that the dark line here is wild, the gray line below is cultivated, much less spiking in the gray because it's not as valuable. But look at this. This is the actual amounts, dry kilograms. And we're seeing a decline. Now, is that because people are less interested in ginseng? No, this is because we're getting wild populations being denuded from sort of bad wildcrafting, right? People just going out and picking out stands or having ecosystems where ginseng flourishes being destroyed. So we've got folks in the United Plant Savers doing some great work along these lines, trying to work on conserving these wonderful American herbs that are being lost due to overharvesting and loss of habitat. So again, the dual pressure of less available and higher cost means you're going to see a lot of adulteration in this marketplace. We're going to just go into extraction very briefly. Again, a lot of these could be an entire lecture, but I'm trying to paint you a picture of the complexity of understanding use and all the factors that go into potentially influencing use. So in TCM, you've got a number of different types of approaches. You've got fresh ginseng. You've got the white Chinese ginseng here. They remove the rootlets from 4 to 6 year old ginsengs and dry them down to get this nice dried root where moisture content is less than 14%. And I find that fascinating that they remove the rootlets. Now, I'm unsure if they get used in the materia medica anywhere or why. Was that simply a process of making the root look more attractive to buyers, or was there a rationale that had to do with clinical endpoint? Red ginseng, which is largely found in Korea, uses the same Panax ginseng, same species, and steams it. And this changes the clinical endpoint. It becomes much more nourishing and is much hotter energetically. And you do see some products available too where it's a fermented red ginseng, which tends to be a wine. This tends to quiet some of the heat in red ginseng and provide some other medicinal endpoints based on their materia medica. Now here's some data from Gaffner et al. Around contemporary extraction effects. And I'll link back to one of these elements in a minute, but you've got a 50% ethanol extract, which is 50% ethanol, 50% water. You've got an ethanol, glycerin, and water extract. And you may know that glycerin is both a way to extract, but it also is a sweetener. And here you have 65% glycerin and the rest water. And you can look over this data if you want in terms of the amounts that you see there. But I'll summarize it down here below where the total saponins are highest in the aqueous ethanol, that 50% ethanol. And they're lowest in the 65% aqueous glycerin. So having alcohol is really good at extracting these ginsenosides. Now, additionally, what happens is these ginsenosides ends up being enzymatically cleaved in the glycerin because they're macerating, they're sitting around, and there are enzymes present from the plant material that in that glycerin appear to be more active. And so what we see over time in storage, and this is in all the extracts, at 25 degrees C, you lose about 13 to 50%, 15% of your saponins. So there, when you go into industry-based concerns and labeling, you have to sort of account for that when you claim how much is going to be in your product a year down the road. Or you need to come up with methods that stabilize these ginsenosides beyond just simple storage. So this last piece around formulation, this really is a whole nother lecture. But in TCM, the ginsenosides, because of their soapy nature, their saponin nature, they're often combined with bitter orange because that thick, soapy quality doesn't really digest very easily. And so the bitter component is often added to help with the digestion and absorption of those compounds. Traditionally, we've always sort of had the raw root that was put into foods or it was powdered. In Western approaches, you have tinctures, you have capsules, but what other forms could we think about? Are there ways to look at nasal-based ingestions of these compounds, particularly if we're dealing with CNS issues and you want direct absorption into the bloodstream in the head region? Expanded use of other ginsengs, right? We haven't gone into all the characterizations of those and how they might be different. And then expanded use with other adaptogens or trophorestoratives and tonics. Now, they all play different roles. Trophorestoratives come out of the naturopathic movement. They tend to be long-term usage. They tend to be very rich in nutritive agents. Tonics come out of a number of different traditions, and they tend to be used in very high doses and oftentimes for short periods of time. And some of them can be used over longer periods of time, but often tonics you have to cycle off of them. And so going back to the concern from Maria, I would look at something like a ginseng being very useful with a number of other herbs that work with nervous system signaling or muscle contraction issues. Kava kava would be one. You could look at a number of different calmatives that work on as nervines that might tamp down that excessive nerve signaling. So these would be ways, again, when we talk about ginseng, we're not talking about it in isolation. There are times it's used as a simple, but more often than not, when we look at we're either looking at their use as a clinical endpoint, meaning one-on-one with a single individual that has a unique signature in terms of what their needs are, what they're facing, and you can construct something that's for them. And even in that case, though, you have primary concerns, others that emerge after primary concerns have been alleviated. So you're looking at some form of stacking function where you have multiple formulary that are constructed over time to resolve a whole set of issues. Or if you're making products, what we're really doing is we're looking at population level issues. And in that case, we can't just look at what it appears in an individual. We have to be able to look and say, how does this condition get measured in a whole population of people? Antoinette, it's nice to meet you. Thanks for showing up. You'll get a copy of the webinar as a video sent out to you via email. Okay, thanks for coming. So these are all things that we can go into in great length, and certainly we do in our program. But I thought I would go ahead and just finish up by saying there's some questions in my mind that need to be refined, right? What are the best growing conditions and timing of harvest for specific endpoints, right? The human gut flora as a biotransformation agent, is it the same in everyone? We know it's not, right? We know that there are very strong differences in people that are both genetic, but also lifestyle based. And what are the role that these multi-constituent ratios play? Again, I pointed out to that by saying, let's compare the 16 and the 5-year. These are things that I'd love to see data around because I think it would help us understand how to apply and use the different species of ginsengs.